the CIBM Center for Biomedical Imaging Breakfast in Science Seminar number 5. It is my great pleasure today to introduce you to our speakers. The second speaker is Pol del Aguila Pla, who is a research staff scientist at CIBM Signal Processing EPFL Mathematical Imaging Section. Pol will be describing the library GlobalBioIm and will be showing how easy it is to formulate imaging as an inverse problem. Thank you, Pol. The floor is yours.
Thank you. Thank you for the introduction. So I've gone for giving a very high-level presentation on imaging as an inverse problem. So I'm working on mathematical imaging on the signal processing side on how to recover images once the data has been acquired. And what I'm going to introduce is not only a very simple framework to understand imaging as an inverse problem, but also a software library that the group developed and that I think can be useful. So my goals here will be basically to present a very simple picture and also to insight, especially within the CIBM collaborations or use of our software. And yeah. So first, for imaging as an inverse problem, fundamentally, this is the scheme that you should picture in your mind when you're trying to think about signal processing methods for image reconstruction. So you have a biomedically relevant quantity, which is usually a quantity that lives over a continuous domain. So it's a density of fluorophores, or we'll see a couple of examples. And then you have to transform that into something you can store in your computer. So that's the first transformation that is relevant to how things will work. Then you have a process, a physical process that happens, or some physics in the instrument that you're using to observe, which we signify always by this letter H. And you have some statistical variability or noise. And there are a few simplifying assumptions that are usually done. So first, we usually assume that the physics are linear, and that this basically means that if you're imaging one object and imaging another object, and then you image both of them together, you're going to get the same as the sum of the two images that you would take separately. As you all well know, probably this is not the case in many instruments, but it's a simplifying assumption that helps a lot. And another assumption is that noise is usually additive. So that means that the statistical variability changes the image that you observe or the quantity you want to measure around an average instead of doing more complicated stuff. Of course, there are very common modalities that don't accept this, but it's usually, again, a valid approximation under some regime. And that usually leads to ill-post inverse problems, which means that many S's, so many of the representations of the quantity you want to have, could lead to the same Y. So that carries you into a problem, which I think Eliana was talking about something similar, which is, okay, you have data, and what happened behind that? Yeah. So the strategy, this shouldn't be here, it's a last minute change. So the strategy to recover an image is really end result. So it hasn't changed for a long time, which is basically trying to find the representation of what you want to know that is more consistent to the data and fulfill certain properties, which we usually call regularization. And the progress, so the progress in the field is measured by two things. So changing H. So here you have changes on the discretization, on the representation of the continuous quantity you want to obtain in the number of measurements, like compressed sensing and so on, and changes in the regularization. So mathematically incorporating knowledge about the solution you want to have. So that you can do with sparsity, you can do nowadays with very popular neural networks, or you can do with better statistical models. So I'm going to start with an example, which is very close to my heart because I dedicated many years in my PhD to work with. And that is specifically relevant to the current situation because it's ELISpot and fluorospot immunoassays. So they are heavily used for vaccine development. So they're actually this machine in the middle is something that came out of the research results in my PhD and is currently being used to try to find a vaccine for COVID. So I'm really happy about that. And it's also a very interesting problem because it breaks the typical intuition of what imaging is, because you basically only obtain in your computer something like this, so a 2D image, but you're actually trying to recover stuff that is 3D. So it's a 2D plus time quantity. So here is a caricaturized picture of what you want to obtain. These are assays in which you have a bunch of cells and you want to know which are the red ones. So basically, which are the ones that are responding to something by generating proteins called cytokines, which you probably know more than I do. And you don't only want to know which ones are generating cytokines, but also with which characteristics over time they're doing that. So basically, you let those cells incubate and then you pass them through one such machine and you get images like that. And I'm gonna do a very high level description of the physics that happen here now. So basically you have these cells generating particles that float around and may stick to the surface, but may also unstick and they keep floating and so on. And the experiment progresses like so for a long time. And in the end, you only get some of the particles that got stuck to the bottom of the assay. And then you image them and you obtain this other thing. So now, how are you going to recover? And this is going to be a more general way of thinking of algorithms for image reconstruction. So how are you going to recover what we want to know from this, especially when the image is much larger than what we saw? So basically, it works like that. You have an hypothesis, you pass it, which we are going to call SI, and you pass it through your model of the physics, H. You compare with what you have. So you basically compute this difference between what you observed and what you predict that you should have observed. And you also compute how good how good your hypothesis is according to your regularization. And then you update and you keep iterating like that. So that's the idea of an iterative algorithm. And then, so if you're lucky and you choose things right, this will happen. And so you'll be able to recover what we want. So now a bit more realistic, the physics of these assays are described by a reaction diffusion absorption process, which looks a bit nightmarish. But basically, once you represent this inside a computer, you can equivalently think of it as basically this S quantity that we want to recover is going to be a set of N images over different times. And what we observe is going to be a sum of these N images convolved, so blurred differently. So here the G1 to Gn are simply different kernels of different width that will blur these images, and you will observe the sum of everything altogether. And then, of course, as I was saying at the beginning, this is going to be an ill-posed inverse problem. And there are going to be many combinations of S1 to Sn that lead to the same image. And we need to somehow choose between those. So here is when you have to choose a regularization in which you basically, what we did was, okay, so there's a location here and there's the time component of this location. And what we said, okay, if there's a cell there and it's generating particles at some point, it should be generating particles throughout the experiment. So mathematically, you formulate this that way. So you basically, your regularizer is a sum of many norms of this kind of vectors across time. So this is for the first example. And yeah, if you're interested, you can read more about it. We will share the slides probably. So the second example that I want to present in this framework is 3D wide field microscopy. I think I don't need to explain what its uses are because there are so many. And in this case, what you're trying to recover is a 3D quantity, which is a density of fluorophore, but you have 3 different fluorophores. So it's a sort of a 4-dimensional object or three-dimensional, but 3 times. And you basically pass it through a microscope. There's some noise process, and then you observe this. So here, again, what can image reconstruction give you? So this is the data that you get, and this is what you can recover by using the framework that I'm describing here. As you can see, there's this is much more usable data than this, and you've probably seen before examples of the convolution. So wide field microscopy, the physics, and I'm not going to pretend I'm an expert in optics, but the basic idea is that you illuminate the whole sample, but when you're observing, you get a sort of a focal effect in which your microscope is focused at only one Z in the optical axis and not in the others. So if your sample is wide, or deep, you will have diffraction limited optics in 3D. So you have to solve the convolution, the blurring problem, and the blur that you're going to observe is going to be of this shape. So basically, it's going to be pretty thin at the middle point, so that you can see this red pane, and it's going to spread out as you go far from the Z in focus. And in order to acquire the data, you just keep changing the Z and you observe a whole volume. But again, you're going to get problems because this is ill post. There are many volumes that could lead to the same image. So how do you choose between them? And this is a very common technique in inverse problems for imaging, which is total variation regularization. So you basically think of the volume that you want to image, and you measure the change of direction in the 3 dimensions, and you try to, among all possible volumes that could explain the data that you got, you choose those that have less variation along these 3 axes. So if we take a Wikipedia explanation in 1 dimension, so basically total variation regularization is trying to recover piecewise constant volumes or functions in one day. And you do this by basically, again, putting a norm and using the gradient, so the computation of these variations as an operator for your regularization. So the point here of the talk is that in GlobalBioIm, we have made a library that makes this very easy. So as the receipt for getting images in biomedical imaging is you learn the physics and then you have an age. And for that, the library includes implementations of most of the usual imaging modalities. So whether convolution, X-ray transform, which is the Radon transform, a generic matrix, what sliced Fourier transforms, sums, selectors of patches, etc. Then you have to learn the math in order to choose one of the common regularizers. So the library includes everything you need to compute a regularizer. And you have to choose what I have not discussed, which is, okay, once I've chosen a problem that I want to solve, how do I actually do that on a computer? And for that, it includes many of the typical optimization algorithms for this. And this is a library written in MATLAB, and it even includes a graphical user interface now. So you basically load your data and you can select from a menu which are your physics, which is the math you're going to use, and which optimization iterative algorithm you want to use. So this is a very simple few lines of code to us to see how easy it is to solve the 3D wide field microscopy problem in GlobalBioIm. And you can see that basically you can afford to write the notation simply as I've written in my slides. So there's H, which is a convolution operator. There's L, which is the gradient in order to do TV regularization later, and so on. So how easy it is to define the cost function, how easy it is to define the regularizer, and so on. And actually, I just left out the bit in which you choose which optimization algorithm you're going to use to solve the problem. There's much more in an ICAS tutorial that we did recently. Here is the link to the CIBM YouTube. And then you can also download the example, the specific example I show snippets of code of in this address. And of course, our library is reachable at that point. So at this point, I just want to acknowledge the contributors of GlobalBioIm, because I basically haven't coded this. I have been a user only. You can get here their contributions from GitHub. The biomedical imaging group, so where I usually work when I'm not confined. Then ERC for the grant that supported the development of GlobalBioIm, from Professor Michael Unser's grant, and then the EPFL. And for slides in this presentation, I would like to thank my PhD supervisor, Joachim Mielden, in the problem of immunohistory. So that is all. Thank you for listening.
Thank you very much.